This is the pre-assignment video lecture for TBL 10, Purine and Pyrimidine Metabolism, presented by Lecture Diekman. The required reading is the clinical correlations in Devlin Chapter 20. A number of the clinical correlations are going to be highlighted during this lecture. However, you need to take a look at all the clinical correlations in Chapter 20. The supplementary reading is the entire Chapter 20. A study guide and a set of practice questions are posted on O2. These are the objectives for TBL 10. You need to be able to describe the biosynthetic pathways for the purine and pyrimidine nucleotides. This includes identifying the source of atoms in the purine and pyrimidine rings. Describe the metabolic control of purine and pyrimidine biosynthesis. You also need to be able to discuss the clinical relevance of purine and pyrimidine metabolism. This includes the disorders of purine and pyrimidine metabolism, as well as the purine, pyrimidine, and folate analog drugs. Please note that you do not need to memorize structures unless otherwise indicated. However, you should be able to look at a structure and identify it as either a purine or a pyrimidine based on its characteristic ring structure. Okay, so what are purines and pyrimidines? The purine bases are adenine and guanine, and the pyrimidine bases are thymine, uracil, and cytosine. A nucleoside is a purine or pyrimidine base that is attached to ribose or deoxyribose. So an example of a purine nucleoside is adenosine, and an example of a pyrimidine nucleoside is uridine. The nucleotides are nucleosides that have been phosphorylated. They can have 1 to 3 phosphate groups. So examples of purine nucleotides include adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate. We ingest purines and pyrimidines in our diet. However, we cannot incorporate these directly into nucleic acids in our tissues. Instead, we have to synthesize purine and pyrimidine nucleotides de novo from amphibolic intermediates. You should also note that all of the pyrimidine and purine metabolism that we're talking about in this lecture occurs in the cytosol. As you have already learned during Molecules to Cells, purine and pyrimidine nucleotides have multiple functions. ATP is an important energy molecule. All of the nucleotides are important components of RNA and DNA. Cyclic AMP is an example of an important secondary messenger. The purine and pyrimidine nucleotides also can be components of coenzymes, or as components of activated intermediates in metabolism. They can also serve as allosteric effectors to regulate metabolic reactions. So let's start with purine synthesis. This slide shows a basic set of instructions for how to build a purine nucleotide. First, you need to get a ribose 5-phosphate molecule from the pentose phosphate pathway. Then, convert it into 5-phosphoribosylate 1 pyrophosphate. This is abbreviated as PRPP, and I'm going to be calling this molecule PRPP for the remainder of the video lecture. Next, you're going to build a purine ring on PRPP by 10 enzymatic reactions in which carbon and nitrogen atoms are obtained from glutamine, glycine, N10-formyl tetrahydrofolate, and carbon dioxide. This will form a molecule of inosine 5-monophosphate, which is abbreviated as IMP. Next, from the IMP branch point, you are going to form molecules of AMP, ADP, and ATP, or GMP, GDP, and GTP. This slide shows the metabolic reaction that converts ribose 5-phosphate into PRPP. This reaction is catalyzed by PRPP synthetase. Mutations in PRPP synthetase can lead to ART syndrome, which is described in Clinical Correlation 20.1. Increased PRPP synthetase activity is one of the causes of gout, and that is described in Clinical Correlation 20.2. PRPP has multiple roles in purine and pyrimidine metabolism. As you will see, PRPP is involved in the de novo synthesis of both the purine and pyrimidine nucleotides. It's also involved in the salvage pathways of both the purine and pyrimidine bases. This slide depicts the 10 enzymatic reactions that convert PRPP to IMP. Please note the roles of glutamine, glycine, aspartate, N10-formyl tetrahydrofolate, and carbon dioxide as carbon and nitrogen donors. Also note that the process involves 2 molecules each of glutamine and N10-formyl tetrahydrofolate. You should also know that some of the enzymatic activities involved reside on separate domains of multifunctional proteins. Please do not memorize all of the intermediates and enzymes involved. The enzymes for which you are responsible from this lecture are listed in the study guide. This figure shows the sources of carbon and nitrogen atoms in the purine ring. Nitrogen 1 comes from aspartate. Nitrogens 3 and 9 are donated by glutamine. Carbons 4 and 5 and nitrogen 7 are from glycine. Carbons 2 and 8 are donated by N10-formyl tetrahydrofolate. Carbon 6 comes from carbon dioxide. You need to be aware that in diagrams such as this, carbon dioxide is sometimes shown as bicarbonate. You need to be able to identify the source of each of the carbon and nitrogen atoms in the purine ring. Please note that sometimes the purine ring may be shown in an opposite orientation. This slide shows the first step in the construction of a purine ring. This enzymatic step is the conversion of PRPP to 5-phosphobeta-D ribosylamine. This reaction is catalyzed by glutamine PRPP aminotransferase. As you have already seen, PRPP is used in multiple reactions of purine and pyrimidine metabolism. Therefore, we refer to this step as the committed step in purine biosynthesis because it commits a molecule of PRPP to the construction of a purine ring. You should also note that glutamine PRPP aminotransferase is the rate-limiting enzyme in purine synthesis. We are going to talk about its regulation in a subsequent slide. Once IMP is made, purine biosynthesis continues on from what is called the IMP branch point. From IMP, 2 branches of the purine biosynthetic pathway produce GMP and AMP. And then from GMP and AMP, kinase reactions produce GDP and GTP, as well as ADP and ATP. Let's talk about the regulation of purine nucleotide biosynthesis. Purine biosynthesis is regulated allosterically by PRPP, IMP, AMP, and GMP. As I've mentioned before, glutamine PRPP aminotransferase is the rate-limiting enzyme in purine nucleotide biosynthesis. This means that it is the major regulated step in the pathway. IMP, GMP, and AMP allosterically inhibit glutamine PRPP aminotransferase, while PRPP allosterically stimulates glutamine PRPP aminotransferase. IMP and GMP act at one site on the aminotransferase, while PRPP and AMP act on a different site. Purine nucleotide biosynthesis is also regulated by the availability of PRPP as a substrate. The more PRPP that is available, the more that the pathway will be driven. Also, GMP and AMP can allosterically inhibit the enzymatic reactions immediately following the IMP branch point. Salvage pathways represent an alternate way of synthesizing purines and pyrimidines. Shown here are some of the purine nucleotide salvage pathways. These pathways can synthesize nucleotides from intermediates in the degradative pathway for nucleotides. So for example, on this slide, we're seeing salvage of hypoxanthine, guanine, and adenine, which are purine bases. Salvage pathways are especially important in tissues that cannot perform the entire process of de novo synthesis. In this process, note the requirement for PRPP. Hypoxanthine guanine phosphoripocyle transferase catalyzes the phosphoribylation of hypoxanthine to IMP and guanine to GMP. These salvage pathways are regulated by their end products. HGPRTase deficiency causes Lesch-Nyhan syndrome, and this is described in Clinical Correlation 20.3. Now let's talk about purine nucleotide catabolism. Guanine and adenine nucleotides from nucleic acid breakdown are degraded to xanthine and then to uric acid. Xanthine oxidoreductase catalyzes the conversion of hypoxanthine to xanthine and then the conversion of xanthine to uric acid. You need to know that xanthine oxidoreductase is the target for allopurinol, an anti-gout drug. Deficiencies in purine nucleoside phosphorylase and adenosine deaminase cause some significant immunodeficiencies, and these are detailed in Clinical Correlation 20.5. So for example, adenosine deaminase deficiencies lead to severe combined immunodeficiencies, which are referred to as SCIDs. This slide shows the first part of the construction of a pyrimidine nucleotide. We start by building a pyrimidine ring by 4 enzymatic reactions in which carbon and nitrogen atoms are donated by glutamine, aspartate, and carbon dioxide. This forms orotate, or orotic acid. This is a major difference between purine and pyrimidine synthesis. In purine synthesis, the purine ring is built directly on PRPP. In pyrimidine synthesis, the pyrimidine ring is made first, and then it's put on PRPP to form orotidine 5 monophosphate, or OMP. Next, OMP is converted into UMP. Step 1 in the figure shows the carbamoyl phosphate synthetase 2 reaction. This is the major regulated step in pyrimidine synthesis. It is also sometimes referred to as the committed step. Deficiencies in orotate phosphoribosyl transferase and/or OMP decarboxylase can cause orotic aciduria. This is described in Clinical Correlation 20.7. This figure shows the sources of carbon and nitrogen atoms in the pyrimidine ring. Nitrogen 1 and carbons 4, 5, and 6 are donated by aspartate. Nitrogen 3 comes from glutamine, and nitrogen 2 comes from carbon dioxide. You need to be able to identify the source of each of the carbon and nitrogen atoms in the pyrimidine ring. Please note that the pyrimidine ring may be shown in the opposite orientation. To continue pyrimidine synthesis from UMP, kinase reactions convert UMP to UDP and then to UTP. UTP is then converted to CTP by CTP synthetase. Well, what about TTP? Don't worry about that yet. We're going to get to the synthesis of DTTP in a few minutes. This slide shows the regulation of pyrimidine synthesis. As I've previously mentioned, the carbamoyl phosphate synthetase 2 reaction is the major regulated step in pyrimidine synthesis. This synthetase is stimulated by ATP and PRPP and inhibited by UTP, UMP, and CTP. In addition, OMP decarboxylase is inhibited by UMP. CTP synthetase is inhibited by CTP to maintain a balance between UTP and CTP in the cell. As you can see on this slide, the pyrimidine nucleotides that are obtained from the breakdown of RNA and DNA are degraded to the pyrimidine bases uracil and thymine. Similar to the purines, pyrimidine nucleotides can also be synthesized by salvage pathways. Uracil and thymine from the breakdown of nucleic acid can be salvaged by conversion to their respective nucleotides by pyrimidine phosphoribosyl transferase. If uracil and thymine are not salvaged, they are subjected to further degradation. Uracil is degraded to beta-alanine, carbon dioxide, and ammonia. Thymine is degraded to beta-aminoisobutyrate, carbon dioxide, and ammonia. You need to know that defects in pyrimidine catabolism are of limited clinical significance because the catabolic end products of pyrimidine degradation are water-soluble and are excreted in the urine. Also, beta-aminoisobutyrate in urine can be used as a measure of DNA breakdown. Purine and pyrimidine deoxyribonucleotides are synthesized from their respective ribonucleotides for incorporation into DNA. ADP, GDP, CDP, and UDP are converted into their respective deoxyribonucleotides by nucleoside 5-diphosphate reductase, also known as ribonucleotide reductase. This is shown in step 1 in the figure. The nucleotide reductase reaction is the rate-limiting step in the deoxyribonucleotide synthesis and requires NADPH. Inhibitors of ribonucleotide reductase are potent inhibitors of DNA synthesis and cell replication. DMP is produced from DUMP by thymidylate synthase. This reaction requires N5-N10 methylene tetrahydrofolate. In step 5, DNA polymerase incorporates the synthesized deoxyribonucleotides into DNA. The next 3 slides summarize some of the clinical conditions associated with defects in purine and pyrimidine metabolism. The relevant clinical correlations in Devlin are indicated. Lesch-Nyhan syndrome is an X-linked disorder that is associated with a severe or complete deficiency of Hg-PRTase. The inhibition of purine salvage pathways causes accumulation of uric acid. A mild Hg-PRTase deficiency can also be a cause of gout. Immunodeficiency diseases can result from deficiencies in purine nucleoside phosphorylase and adenosine deaminase. Adenosine deaminase deficiencies lead to severe combined immunodeficiencies, also known as SCIDs. Tumor lysis syndrome is actually a side effect of cancer treatment. Patients undergoing cancer treatment can show increased serum and urine uric acid levels due to the degradation of nucleic acids and nucleotides from destruction of tumor cells. Allopurinol is frequently included in treatment protocols to limit uric acid buildup. Deficiencies in orotate phosphoribosyltransferase transferase and/or OMP decarboxylase can cause orotic aciduria. This disease can be successfully treated by feeding with uridine. Gout is characterized by high levels of uric acid in the urine and blood. High levels of uric acid in the blood is also called hyperuricemia. The metabolic defects in gout have not been completely defined, but include increased PRPP synthetase activity and a partial deficiency in Hg-PRTase. Increased PRPP levels can cause increased flux through the de novo purine synthesis pathway, and this leads to increased uric acid levels. Decreased Hg-PRTase activity prevents salvage of hypoxanthine and guanine. This also can lead to increased uric acid levels. In addition, dietary purines can contribute to hyperuricemia. Crystal deposits of sodium urate, called tophi, form in joints and soft tissues. These can cause acute or chronic inflammatory reactions, which then can lead to acute gouty arthritis or chronic joint destruction. And just when you thought we were done talking about the disorders of glycogen metabolism, we have to talk about von Gurke disease again. Gout can result from von Gurke disease, which is a deficiency in glucose 6-phosphatase. The elevated glucose 6-phosphate levels increase flux through the pentose phosphate pathway, which increases production of ribose 5-phosphate. The elevated ribose 5-phosphate levels drive the synthesis of PRPP. The elevated PRPP then drives the de novo purine synthetic pathway, leading to increased uric acid levels. And the increased uric acid levels can then cause gout. The next 2 slides discuss the use of purine and pyrimidine analogs as drugs. Allopurinol is a purine analog that is used in the treatment of gout. It inhibits xanthine oxidoreductase and reduces the production of uric acid. Cytosine arabinoside is used in the treatment of multiple cancers, such as leukemia. It first must be metabolized to its ribonucleotide triphosphate form to be effective. It then inhibits DNA strand elongation. 6-mercaptopurine is a purine analog that is used as an anti-tumor drug. HGPRTase metabolizes it to its ribonucleotide monophosphate form, which then inhibits the production of PRPP by glutamine PRPP aminotransferase. It also inhibits the conversion of IMP to AMP and IMP to GMP. 6-mercaptopurine is also a substrate for xanthine oxidoreductase. So 6-mercaptopurine is frequently given in combination with allopurinol to prevent its conversion to uric acid. Purine and pyrimidine analogs are also used in the control of some viral infections. Acyclovir is acycloguanosine, which is a purine analog. Acyclovir is used in the control of herpes infections. Acycloguanosine is converted into acycloguanosine monophosphate by HSV-specific thymine kinase, which is only present in infected cells. The resulting acycloguanosine triphosphate is incorporated into the replicating viral DNA chain, and this causes DNA chain termination. AZT is a pyrimidine analog that is used to control HIV infections. AZT is phosphorylated to AZT triphosphate, which blocks HIV replication by inhibiting HIV DNA polymerase. Purine and pyrimidine analogs are not the only drugs that are used to disrupt purine and pyrimidine metabolism. Antifolates are used to inhibit reactions that require tetrahydrofolate. Methotrexate is a folic acid analog that is used in the treatment of multiple cancers and rheumatoid arthritis. Methotrexate inhibits dihydrofolate reductase. This prevents the generation of tetrahydrofolate. The decreased levels of tetrahydrofolate inhibit the de novo pathway of purine synthesis, as well as the synthesis of DTTP. This is the conclusion of the TBL 10 pre-assignment video lecture. Please be sure to read the clinical correlations in Devlin. I have highlighted a number of them for you during this video lecture. The study guide should help you understand the level of detail for which you will be held responsible for this lecture. A set of practice questions is posted on O2, and I suggest you use them in your preparation for the TBL quiz and for the exam. Thank you, and please let me know if you have any questions.